Electric fish are strange creatures. These real-life Pikachus, the cute Pokemon character that can store electricity, actually have specialized organs that can create large electric fields. Different species produce different amounts of electricity. Strongly electric fish, like the infamous electric eel, use their powerful discharges to stun prey or defend themselves from predators, while weakly electric fish, like the ghost knifefish of the Apteronodidae family, use gentler discharges to sense their environment or communicate in murky or dark water. In most cases, the fantastical organs that help these fish survive evolved from mussels. Most electric fish create their signals by co-opting normal processes involved in muscle movement, like the electric discharges from sodium channels. Sodium channels are tiny, tightly controlled pores that control how positively charged sodium ions enter cells, creating a current. As they evolved, electric fish lost a sodium channel in their muscles, moving it to their electric organ, allowing them to discharge electricity and send out signals without causing their muscles to twitch. This crucial change is one of many remnants of a genetic hiccup early in fish evolutionary history that duplicated every gene. That is to say that at some point, ancient fish made a second copy of every gene in their genome. This gave fish extra DNA to play around with and alter, like having a spare car in your driveway to tinker with, but genetics alone doesn't explain the diversity of electrical signaling languages in electric fish. Some fish send out brief pulses of electricity, with long pauses between them, similar to sonar on Navy ships. Others discharge huge, lethal voltages when threatened. Ghost knifefishes, Apteronodidae, are particularly curious, generating an incredibly fast, continuous electric discharge. But their supercharged system isn't the only unusual thing about these fish. Their electric organ evolved from their spinal cord instead. Intrigued by this oddity among oddities, a group of American researchers combined gene sequencing, evolutionary analysis, and biophysical experiments to find the genes responsible. Wondering how knifefish may have modified their sodium channels, the researchers found that, in addition to the genome duplication event common to all electric fish, Apteronodidae also duplicated a second sodium channel gene, SCN4A. The researchers estimated that the newly duplicated gene started to express in the spinal cord within 2 million years, a breakneck pace in evolutionary terms. An extra gene coming from this second duplication, SCN4AB1, now makes 20 to 45% of all sodium channels in the knifefish's electric organ. This is the first example of a muscle sodium channel gene appearing in a non-muscle tissue, but it wasn't the only thing that made Apteronodidae unique. The order of amino acid building blocks that make up the sodium channel protein also changed. Such amino acid modifications can have dramatic effects on a protein's function. For example, small changes in amino acid sequences are responsible for both cystic fibrosis and sickle cell anemia in humans. Illustration of 4 fish Wikimedia Apteronodidae actually had many more mutations than expected by random chance. A finding like this is really exciting for evolutionary biologists. Natural selection generally purges harmful mutations from the gene pool, allowing only harmless or beneficial mutations to reproduce in the next generation. To find out whether the changes in the sodium channel's amino acid sequence changed how the protein functioned, the team took a human sodium channel, modified it, and expressed it in frog cells, which are easy to mess around with in the lab. Using this model channel, they were able to run a number of electrophysiological tests, exploring how the changes affected the electric